Wonderful. Oh, well, thank you everyone for sticking around. And it's a real honor for me actually to speak after, oops, oh, this is not a timer. After Julia, whose work I've followed for a long time. And what I really appreciate about the work of her and her collaborators is taking a very detailed molecular genetic approach to thinking about these whole plant traits, which is a really hard thing to do. And one emerging theme, I think, from Julia's work and also from that of the other sort of best plant environment interactions is thinking about the challenge of living in an environment that involves a lot of trade-offs. And people in the plant field have considered the challenge of trade-offs for a long time. So classically, ecophysiologists have considered the challenge of acquiring some resource, and then once you've acquired that resource, making some decision about how to invest it. And it could be invested in any different ways, such as growth, defending against pathogens or stress, and other kinds of stress, storage. I think today we probably would also include exudation of volatiles in this equation, but ultimately in reproduction. And each one of these arrows involves, as I suggested, a trade-off. And it's a series of trade-offs that plants have to optimize in order to survive in the environment, or in our case, optimizing plants to provide resilient yield. I am particularly interested, and I'll talk today about this really tight interaction between acquisition of resource, particularly carbon, and then growth and maintenance. And I think this is a really fascinating trait because in the sense that resources may be limiting, acquisition is affected and growth is affected by the environment. And yet plants can make decisions about their growth patterns that could then help modulate those challenges, right? So if if you're short on nutrients, you could expand your root growth, for example, okay? And I think what this work really highlights is that there's a very tight and really unbreakable connection between plant physiology and plant development. Now, as I mentioned, these are whole plant traits to consider. And these become very difficult to study using reductionist approaches. And so the approach that we've used to understand growth rate and its control is to measure growth rate as RGR. So now we're talking about individuals, right? We heard RGR in the context of populations the other day, but relative growth rate measures the daily gain of plant biomass as a function of that plant's mass on that day. So this allows us to make comparisons among genotypes, among species, sort of on a level footing. And again, dating back 30 or more years, plant physiologists have considered this very complex trait, relative growth rate, as a function of three other fairly complex traits. One has to do with the structure and composition of leaves. So basically, how is the photosynthetic apparatus deployed? The proportion of the total plant biomass that is invested in leaves, so it's invested in photosynthesis potentially, and then the rate at which carbon or mass can be acquired through these tissues. So we multiply all these things together and we get relative growth rate. And again, each one of these is a complex trait, but they're more readily studied, again, in a comparative framework than, for example, just an entire plant's growth rate. So the questions that I'm focusing on in this particular project are big ones. What controls growth rate differences within and between species? And then the broader context of this is thinking about sort of evolution, but also plant breeding, and how has this control of growth rate been manipulated by plant breeders, which it certainly has indirectly, if not directly, but also in evolutionary context and thinking about how plants are evolving in response to climate change. So I'm just going to focus on this first one today, sort of a mechanistic story. And our model for studying this is, as Jeremy mentioned, Brachypodium. This is a model grass species. Just a quick overview of it. There's a couple sort of beautiful old classic drawings of Brachypodium. There's a worldwide distribution of Brachypodium. The model species most of us work on is Stachyon, which is circum-Mediterranean right here. And the real appeal of Brachypodium as a model is its phylogenetic position as the sister lineage to two big groups of very important grasses for us. One are POA and the many forage grasses, but also important cereal crops like wheat, oat, and barley. And the other real power of it is the tremendous synteny we see among grass genomes. So work that is identifying genes or functional elements in Brachypodium, you can often then go in other grasses and find orthologous regions. The JGI has for many years invested heavily in Brachypodium genomics. I've been involved in some of that over the past 6 years, and just summarizing some of those available resources, but I just want to make the point that this is really now a model genus for plant genomics, plant evolution, plant physiology, because of the genomes we have across the genus that are available, as well as things like mutant populations, recombinant populations, and so forth. So to get to the question of today, what controls growth rate differences within and between species? Again, we're going to try to decompose relative growth rate into these constituent traits. And the way we went about that was using undergarnomics to spend a lot of time cutting up plants into their constituent parts. So we grew a big diversity panel of 2 species. I'll show you those in a minute. For the first 7 weeks of growth, so this is basically vegetative establishment. Plants did not flower during this period. And by chopping the plants up in all sorts of little bits, we were able to directly measure relative growth rate, specific leaf area, leaf mass ratio. This is a profoundly hard thing to estimate accurately, but since we have 3 knowns and 1 unknown, we could calculate this one with these guys. What do these look like? Well, so we looked at 2 different species. We measured 6 inbred natural accessions of Brachypodium sylvaticum. This is a perennial model that's been developed by John Vogel's group, as well as 8 inbred accessions of the annual Brachypodium dactylicum. This is sort of the reference species for the genus. You can see here that there's a significant difference in growth rate between this annual and this perennial. That's a whole nother story that I'm not going to tell today, but there's a lot of really interesting things to be told about annual and perenniality in this genus. But just to make the point that you can see differences among accessions, each point is a separate accession, but also differences between the 2 species. Okay, so we have genetic diversity in relative growth rate. That's great. This is the sort of the starting point for understanding function in our approach. Looking at the different components of RGR, we can see cases where we have significant within species diversity in SLA, though the 2 species don't differ, significant genetic diversity in leaf mass ratio, and so on and so forth. Okay, so again, this really just provides us with lots of natural variants to then try to understand how these, what might be driving these phenotypic differences we see. I want to focus on how we're using this genetic diversity to get down to function. And by simply asking at this point, what are the transcriptional drivers of growth rate variation within species? And the approach that we've used in this case is transcriptomics, where we took the roots of 4-week-old plants. So these are sort of in the middle of that growth phase I talked about. We extracted RNA, we sent the RNA out here. The JGI did a huge amount of RNA sequencing as part of this project. And what this allowed us to do is to estimate gene co-expression networks using traditional WGCNA approaches. So we're reconstructing, hypotheses about co-expression modules. And because we have these, in this case, 6 different inbred lines, we really have sort of 6 biological entities. And so we can assign each one of those accessions a value of a module eigengene. Okay, so this is really decomposing expression variation in many thousands of genes into these smaller manageable traits. What we then do is, because we have all this wonderful phenotypic data, we can look for correlations between our phenotypes, in this case, leaf mass ratio, and the module eigengene, where each one of our accessions has a value of LMR, some module eigengene value, and then we can look for regressions. And these are genetic correlations. And just as one example of that, here's one case where, if you know WGCNA, they have these silly colors. So the salmon module shows a significant correlation with leaf mass ratio. So what is that really? How is that useful? Well, really all this is a set of transcripts that show a genetic correlation with this really cool trait of leaf mass ratio. Okay, but this represents a hypothesis, right? This is just the starting point for functional genomics. So in order to test those hypotheses, we performed a second experiment. This time, rather than using genetic diversity, we're going to use a treatment, environmental treatment, to impose some phenotypic diversity in expression. And what we did is, because we're interested in growth and carbon assimilation, carbon allocation, we modified the amount of carbon available to these plants by changing the amount of CO2 in the growing environment. So these are in growth chambers where we scrubbed out CO2, so it's carbon limiting. We have a control, and then we have a carbon excess treatment, a very, very high CO2. So not mimicking carbon, not mimicking climate change, but trying to, again, to perturb the carbon budgets of these plants. Okay, and again, we extracted RNA. So I didn't show you that there. We extracted RNA. There's an arrow down there. It went away. From all these. And just as a check to convince you that we have actually manipulated these plants, we performed gas exchange analyses using Lycor machines. So you can see that for the 3 species we included in the study, at increasing CO2, we see this increasing in carbon assimilation. So if you give them more CO2, they're going to take up more carbon. So they have this extra source of carbon at higher CO2. And you can see this at a whole plant scale with this net assimilation rate that I mentioned earlier. So what do they do with all that extra CO2? I showed you the big arrows, different things a plant could do with some acquired resource. Well, here we're looking at relative growth rate, this big trait. And what I think you can see is that this one perennial species, Brachypodium sylvaticum, as you increase from sort of ambient CO2 to very high CO2, we see this massive increase in relative growth rate. So in the case of the perennial, it's taking that extra carbon and it's growing more. It's growing faster. It's becoming bigger. Okay, and the proximate cause of this actually is an increase in tillering. So this is now a project we're looking at to try to understand, how is it exactly it is that this perennial is increasing its tillering rate, where we don't see that effect in the annuals. Again, that's a second story to tell. But what I want to do now, there's my RNA-seq, is to use these manipulations to then test our hypotheses about the transcript associations we see with, in this case, leaf mass ratio. So again, recall that we have this diversity panel where we identify this correlation between particular co-expression modules and the trait leaf mass ratio, right? So that's using genetic diversity. And then we also have the second experiment where we're doing this environmental treatment. So now we have the CO2 manipulation, and we could look at genes, for example, that show a significant expression response in one species that don't show a significant expression response in the other species, or they show differing patterns of response. I want to just highlight a couple examples out of here. So in this box over here, these are cases where in Brachypodium sylvaticum or perennial, we see strong upregulation of genes that are in fact downregulated in distachyon at high CO2. There's 164 genes that show that pattern. Interestingly, a very large fraction of those genes are in this module that I highlighted earlier, this so-called salmon module. Don't ask what's in that module, it's just a bunch of genes. And what's really cool is that one of those genes, in fact, one of the most highly connected genes in that module is a Werke transcription factor. We just heard about Werke a minute ago. It's a hub in this module, and it's very, very strongly increased in expression in the perennial, but not in the annual. Okay, so again, we're using this genetic diversity, these environmentally induced differences as a way to identify candidate genes. We're now actually collaborating with Sam Hazen, who has DAP-seq and ATAC-seq data, again, from another JGI CSP, to try to dive into some of these candidates and study them in a more functional way. I'll just briefly highlight another really interesting response we see. So while the perennial increases relative growth rate, the annuals store a bunch of carbon in their leaves. So we give them more carbon, they're fixing more carbon, and for whatever reason, they're stashing it in their leaves. They don't seem to have either the ability to export this or to dump it someplace else. So we see a huge increase in starch. In this Stacyi, we see a big increase in sucrose. I'm not showing you here in distachyon. And those leaves actually get considerably thicker. They have higher SLA. Okay. I won't go into the details of this today, but we're using the same approach here, looking at net assimilation rate, and the correlate, it's a good correlate of gas exchange for carbon assimilation, and using the same approach I just outlined to identify, again, transcripts associated with these traits, and to try to dive down using this comparative approach to find potential candidates. Okay, I went super fast. That's good. So what have I told you today? Well, this is just a little piece of this broader project, looking at the physiological and transcription control of growth in grasses. And I hope I've convinced you in passing that growth rate really is an important component of plant-environment interactions. It allows plants to acclimate to the environment to maximize their fitness in different environments. Brachypodium species show tremendous range of growth rates, both within and between species. And in response to this excess photosynthate we've given in one experiment, we see a really sort of strange pattern where the perennial has this really high growth rate, the annual is putting a bunch of carbon into storage, and we're sort of trying to tease out some of those exponents. I want to finish just by acknowledging the folks who did this work and also make a real strong plug for the JGI CSP program. All the RNA sequencing I've shown you today was done as part of the CSP that I had funded actually before I started my lab at MIT, and it really was formative in getting my lab up and running. A lot of that data became pilot data for now funded grants from NSF and others. So the CSP has been really critically important, and I think you've seen actually there's at least 3, maybe 4 different CSPs have been represented in the work I talked about today. So I'll recognize sources of funding, folks who did all this, and if there's time, I'd be happy to take any questions.
Thanks, Dave. Let's take one question before the break, and I know we're in the break, but we're getting there. Does anybody got a question for Dave? Oh, see, now I said break, so no one has a question. Dave, so the response in the perennial was pretty amazing, for CO2 accumulation. But I'm kind of more interested in the question of why is Stacyi stashing starch at higher CO2? Do you got any?
We don't know. I mean, the hypotheses, well, so there's a longstanding hypothesis that annual plants might be sink limited in the sense that they, because they're so focused on rapid growth, reproduction 1 year, they don't have a lot of plasticity and where they could sort of put extra resources. They would, in principle, invest in reproduction, which we don't measure here. So, mechanistically, I can see 2 reasons. One, they are sink limited. Whatever developmental response that the perennial is showing, like making more tillers, whatever, they just don't have that response. They don't have extra merostems. The merostems are non-responsive, something like that. The other could be just a simple export thing. It could be that there's all this, photosynthate in the leaves, and they can't export it fast enough. So we're trying to explore both of those hypotheses now.
Yeah, thank you. Super neat. Okay, let's thanks Dave again, and catch up with him in a break. Thank you.